HEERANYAA NADARAJA and I am from TPT 4A. Today I would like to perform on my practical test 2, which is Explant culture. Videos. Here I am having detergent, distilled water, 70% of the ethanol, 50% of the Clorox, twin 20 solution, isolate, and I'm having a Bunsen burner. This candle we are assuming has a Bunsen burner. Having 2 beakers. This beaker will help us to place our explant. I'm having a chop board. Usually we use a metal chop board for the laminar airflow activities. So I'm replacing this chop board with that. And then I'm having a scalpel, a faucet, and a holder for the scalpel and faucet. And here I'm having 2 containers with the agar, which represent the 2 petri dish with their media culture. And here I'm having a glass jar, which is represent the glass jar that we use for the media for the explant culture. And this straw I'm represent as a dropper. This is our explant. Now we are going to take out all the leaves from this plant, and we are only taking the shoot, about 3 centimeters of the shoot of this plant for our explant culture. We can take any kind of plants for this experiment. So here we go. We're going to take about 3 centimeters of this. Now I'm going to rinse the shoot using the detergent and running tap water. Now I'm going to place here. Let the tap water to run for 15 minutes. After 15 minutes, the shoot is now ready to culture. After 15 minutes of switching on the UV light of this laminar airflow cabinet, we will switch off the UV light of this laminar airflow cabinet, and then now we are going to wipe the laminar airflow cabinet using 70% of the ethanol. So this is a sterilization process to reduce the risk of the contamination of the outcome of this experiment. So we should sterilize our working place before the experiment starts. Wiping the cabinet with the 70% of ethanol, we are going to use the same 70% of ethanol for us to wipe our hands. Letting the laminar airflow to be dry, now I'm going to light on the Bunsen burner. So this candle we are assuming has a Bunsen burner. Before placing the beaker that contain our explant material, we should wipe or spray the beaker using 70% of the ethanol. So here, the Bunsen burner. And then now we are going to pour about 50% after 10 minutes to clean the Chlorox solution inside the Explant. Now we are going to sterilize the holder that are going to hold the forceps and the scalpel. They also can be sterilized using this alcohol or can be sterilized using the Bunsen burner into the low flame. And now we're going to take out the faucet. Usually in laminar airflow in the lab, the faucet will be covered by the aluminium foil. So we can assume this tissue has aluminium foil. So we take out the cover and then we will be start sterilizing this faucet using the Bunsen burner into the low flame. So this is known as the heat sterilization. We are going to take out the scalpel. We are going to sterilize the scalp before the use. Having the chop board, usually in lab we will have a metal chop board which is covered by the plastic. So I'm representing this wooden chop board has the metal chop board. With the alcohol. Again, we're going to sterilize the faucet. We're going to take out the shoot of this plant, the up one. And then take it down back. Make sure the forceps should hold the shoot in 90 degrees. So here you go. Then I'm going to place it. So this is our glass jar that contain the Explant.